Hello and welcome back to Biology for Beginners. As always, I'm your host, Morgan. In the last video, we completed our discussion of Chapter 10, which was all about genetic material and how the information in that genetic material goes from DNA to RNA to the eventual expression of that gene in the form of the protein synthesis. Today, we begin our walk through Chapter 11, which is all about how genes are controlled. So the first thing we need to talk about is how gene expression is actually controlled. So today I bring you part 1 of the control of gene expression. In science, when we want to start talking about a new topic, we always start with something simple. Since this is biology and we're talking about the control of gene expression, let's begin by talking about prokaryotes. And the prokaryote in question that we're going to discuss today is E. coli. Now, E. coli is obviously pretty infamous because these little things can exist on the food that we eat, and if things aren't prepared properly, they can get in our body and make us very, very sick. But if we think about it, the environment on that food and the environment inside our bodies are very, very different. So the question we need to ask is, how can E. coli cope with those changing environments? Well, the answer to that question is through gene regulation, which is essentially turning genes off and on when they're needed or not needed. Kind of like how the light switch in your house turns the light on and off when you need or do not need that light. And before we move on here, recall that gene expression, which is that light switch being turned on, is that flow of information from DNA to RNA to that protein or polypeptide. The first thing we need to talk about today is something known as the operon. And the operon is basically a stretch of DNA containing all of the genes necessary for a related function. The specific operon we're going to talk about right now is the lac operon, and this contains the genes necessary to produce the enzymes to process lactose in that E. coli. Now, lactose is the sugar from dairy products like milk, so if you have E. coli hanging out in your gut and you drink a glass of milk, that E. coli needs to turn on this operon so that the genetic information that's contained on it can be moved from the DNA to RNA to the protein enzyme so that that lactose can then be processed. So besides having the specific genes that code for those enzymes, the operon also has the promoter, which if we think way back to our Chapter 10 discussions where we talked about that flow of genetic information, we know that the promoter is actually the sequence of DNA that lets RNA polymerase know that this is where I need to attach and start transcribing that code. But between the promoter and the genes that need to be transcribed is something known as the operator. And the operator is basically the switch that turns on and off those genes. And at the operator, we have a repressor that will attach. And if we note that repressor kind of stretches out over the promoter a little bit, so RNA polymerase can't actually attach when the repressor is bound to that operator. And so there's also something that's not part of the operon known as a regulatory gene. And this regulatory gene is usually located just outside the operon, and that is the piece of DNA that codes for the repressor. And that particular gene is always going to be expressed. So it's never turned off or on. Those repressors are always being made. So this is just kind of that image blown up a bit so that we can see on the left side in the dark blue is that regulatory gene that codes to make that little orangey reddish repressor. that attaches there at the yellow operator and extends a little bit over to block the RNA polymerase from attaching at the green promoter. And then in the light blue, we can see the lactose utilization genes, which are the genes that code to make the enzymes that process lactose. So when lactose is actually present in E. coli, it can then bind to that little space that we can see on the repressor and change its shape. And once the shape of the repressor is changed, it can no longer bind to the operator, which then means that the promoter is completely free for RNA polymerase to attach, and the operon would then be turned on because the genes can then be transcribed and the flow of genetic information can then continue from that DNA to RNA to the proteins necessary to break down the lactose and utilize the energy stored within it. So this is just a bigger picture of what we saw on the previous slide. Again, we can see that lactose being present, binding to the repressor, and then RNA polymerase being able to attach and do its job to create that RNA, and then the enzymes can then be produced from that strand of RNA. Now, once this whole cycle keeps going, eventually the lactose will no longer be present, it will no longer be bound to the repressor, and the repressor can then reattach to the operator and the gene will be switched off again. Now we need to kind of discuss the other types of operons, since the lac operon that we have just talked about is only one kind. And the lac operon is known as an inducible operon. And this is because it's normally turned off, but under the right conditions can be switched on or induced. So if it's normally off, but under the right conditions can be turned on, the operon is known as inducible. The other type of operon that we see pictured in this table here as an example is the TRP operon, which is known as repressible. Now, it's normally going to be switched on, so the normal state for the repressor is going to be the inactive state. However, in the presence of tryptophan, the repressor will then bind to tryptophan and be made active. And once it's made active, it can bind to the operator on that TRP operon and switch the whole operon off. So once again, if the operon in its normal state is off, it's inducible. If in its normal state it's on, it's repressible because it can have some sort of cue and then be turned off or repressed. The other thing that we're going to talk about right now that's not shown in the tables on this slide is something known as an activator. Now, activators are these little molecules that will bind to DNA and actually stimulate transcription. So they make RNA polymerase want to attach or make it easier for RNA polymerase to attach and basically turn on the genes at that point because it kind of forces them or makes it easier for them to be expressed. So activators bind directly to the DNA and stimulate the transcription of the genes in that area. Now, this brings us to eukaryotes like you and me. And while there are single-cell eukaryotes, all multicellular beings are eukaryotic, and that brings us to some more challenges when it comes to gene regulation, in that all cells in a multicellular being have the same genes present because they have the same DNA. But different cells express different genes. And the way they do this is through differentiation, which is basically the process by which cells are specialized or how cells have a career given to them. So it's the thing that takes that stem cell and makes it a kidney cell, bone cell, skin cell, whatever. All those special types of cells in the very beginning came from the undifferentiated stem cell as a base, and then eventually got their little cell career and were able to reproduce and produce more of that cell type. So the first thing that kind of helps control genes that we need to talk about in these multicellular eukaryotic cells is DNA packing. Now, if we recall, DNA in each human chromosome is thousands of times longer than the nucleus diameter. But how the heck is this possible? Well, it's possible because the DNA will actually wrap around these small little proteins called histones and form sort of a bead or a nucleosome. So it'll kind of become this like beaded string-like thing with the DNA wrapped around the histone, forming the nucleosome. And the nucleosome specifically is DNA wound twice around a core of 8 histones. And then coiling will continue, and then it results in what we know as that lovely little chromosome that you can see in the image here on the slide. And one important thing about DNA packing, because things are coiled so tightly, it tends to block gene expression because it's kind of difficult for RNA polymerase to get in there and begin that whole process of going all the way from DNA to protein. So genes within those more compacted chromatin are less likely to be expressed. And as we can see here, this is just kind of a bigger picture of what we saw in the previous 2 slides, with the DNA wrapping around those histones, forming the nucleosomes and continuing to coil and form the chromosomes. Another control of gene expression here is something known as epigenetic inheritance. And I know that is a very big, scary sounding term, but it's not as complex as it sounds, I promise. So epigenetic inheritance is basically an inheritance of traits by mechanisms not directly involving that nucleotide sequence. So we know that the nucleotides are the actual little pieces that make up the DNA and make up the code for the particular genes to be expressed in the organism. Well, there is a way to kind of modify that without changing around the nucleosomes or even changing the DNA. And because it's not changing the DNA, epigenetic inheritance is not permanent, and it may actually be reversed. And it's not really necessarily inherited from parent to offspring, but from cell to cell. Now, that being said, epigenetic inheritance is actually still a big field in the world of biology research, and we are still learning a lot about it. But one way this happens is through the methylation of DNA bases. And what happens here is that a methyl group, or CH3, will be added to the base on DNA, usually cytosine. And this methylation does not actually change the nucleotide sequence. You just have this kind of extra methyl group hanging around. And because this is there, it will still go through the duplication process, and this methylated DNA can be passed on to new generations of cells. And the current research is kind of showing that this whole process may actually play a role in turning genes off. So this is, again, like a fairly new field of research, and we're still trying to figure out how it works. But we've also have evidence that shows that improper or insufficient methylation of DNA can lead to problems within the organism, especially in the embryonic phases. But again, this is still a field of biology that has more questions than answers, and we are still exploring this whole realm of epigenetic inheritance. The next form of gene expression control we're going to talk about is known as Inactivation. And this particular control mechanism occurs in female mammals. Now, female mammals do have 2 X chromosomes, so one of those X chromosomes can be rendered inactive. Therefore, the genes on that specific X chromosome will not be expressed. And one way to actually demonstrate how this is happening is through lovely little tortoiseshell cats. And tortoiseshell cats are the ones that have the black and orange splotches all over. The reason that we're looking specifically at tortoiseshells and not the calico is because there's not as much white. What's going on with white patches on calicos is a whole nother level of gene expression that we're not talking about today. So for this discussion, the tortoiseshell with the lovely black and orange coloration. Now, for this X inactivation to happen, one X chromosome will be rendered inactive, and this occurs completely at random. And this is done by the X chromosome being condensed down really, really small. And that condensed X chromosome is known as a bar body. So again, let's move over to our lovely little example here, which is the tortoiseshell cat. In the tortoiseshell cats, the color for the orange and the black are carried on the X chromosomes. And the tortoiseshell cat has one allele for orange on one of the X chromosomes and one allele for black on the other X chromosome. So in the orange patches, we see that the X chromosome carrying the allele for the black fur is rendered inactive, and in the black patches, the X chromosome carrying the allele for orange fur is rendered inactive. So blowing up that picture a little bit here, we get a little bit of a more clear view. As we can see in the early embryo, both of those chromosomes are going to be the normal length. However, through the processes of cell division and duplication, at random, one X chromosome is going to be condensed down into a bar body, and because as the cell is duplicated, those 2 daughter cells can end up with slightly different genetics there, in this case being which chromosome is rendered inactive. We get these little populations of cells that carry either the active chromosome being the one carrying the gene allele, or the active chromosome being the one carrying the black allele. And so it results in this lovely little orange and black patchy look. So just by looking at the cat, you can know which chromosome is active in which areas based on the color of the fur there. So how exactly can transcription be controlled in these eukaryotes, especially that the chromosomal DNA needs to be unpacked so that transcription can happen? Well, in general, in eukaryotes, the default for a gene is to be off. Now, of course, there are some genes that the default will be to be on, but the majority of genes are going to be switched off. And so things like activators are very important as opposed to things like repressors, like what we talked about in the operons and E. coli. So eukaryotes, activators are more important than repressors since the default is for the gene to be off. And because of this, we need to discuss what transcription factors are. And transcription factors are basically the necessary components to trigger transcription to happen, specifically the activator proteins and the other proteins that are necessary to make things kind of move in the right way so RNA polymerase can get in there and do its job. So what happens when those activator proteins are present is that they're going to bind to things known as enhancers, which are little parts of the DNA, usually pretty far away from the gene that's about to be transcribed. And they're basically there just for those activator proteins to bind to. And once the activator proteins do bind to the enhancers, what's known as the DNA bending protein can get in there and literally bend the DNA around so that the other proteins can bind with those activator proteins and kind of stick on to the promoter and pull it into a configuration where RNA polymerase can attach, and that gene can then be transcribed and the gene can be expressed like it needs to be. And the last thing that we're going to talk about today is known as alternative chromosome splicing, because once that gene has been transcribed, recall that the RNA that results has things called introns, which are the non-coding parts of the RNA that must be removed. And then it has the exons, which are the little bits of the RNA that actually have the coding parts and need to be then spliced together. Well, RNA can actually be spliced together in multiple ways. And the result here is that you'll have one gene essentially coding for more than one thing. And it's not as confusing as it sounds. It's literally just either you'll get one exon instead of another, and it kind of just depends on what is needed by the cell at that time. And one interesting thing here is that more than 90% of the human protein coding genes can actually undergo alternative splicing, and our DNA can actually code for many, many more proteins than there are genes held upon that DNA. All right, well, I know it's been a little bit heavy today, so this is where I'm going to leave you for the moment. Next week, we'll have part 2 of this discussion, but I do hope you have a wonderful rest of your day, afternoon, evening, what have you. I know that this chapter is going into more things than your average entry-level biology class will go into, so I do apologize if the information is a little bit more heavy than what any notes from class might tell you. I'm just going by what's in the book, although most entry-level classes do omit a lot of things. So the primary things that you'll probably need to focus on are going to be the things that have to do with the eukaryotes, like the X inactivation and the other gene control in the section about eukaryotes. So that being said, I hope you found some value here. If you like what I'm doing here with my little educational videos, my environmental stuff, my occasional rants when I just have to talk about something and spew those thoughts out into the internet that are usually tangential to outdoors, science, and the environment, please consider clicking that subscribe button, perhaps give me a thumbs up, and as always, please leave me your thoughts down in the comment section. I do enjoy interacting with commenters, and I will see you all in the next one. Thank you again for watching this video. Don't forget to like, comment, subscribe, and ring that bell if you would like to see more. And if you'd like to follow me on any of my other social medias, the links are down in the description below. Don't forget to check out thereptilegoth.com for all of my articles and blog posts. If you found any value in this video and you would like to help support the channel, please check out my Patreon page. That link is also in the description down below. And a special thanks goes out to my Diamond Dragon patron, Diane V. What you're doing is really helping me fund a dream here. I will see you guys all in the next one.